Imagine discovering that every living thing on Earth is built from a single universal code. A code only 4 letters long. A code that determines your traits, your risks, your strengths. A code that can be read, copied, and eventually rewritten. This is the story of how scientists uncovered the blueprint of life, and how that discovery launched the age of biotechnology. Welcome back to Scientific Careers. This is episode 6 of our 10-episode journey through the history of the pharmaceutical and biotechnology industries. Today, we explore the DNA revolution, the moment biology became information. Let's begin. By the early 1900s, scientists knew traits were inherited, but they didn't know how. They didn't know what carried information. They didn't know how traits passed from parent to child. They didn't know what controlled the machinery of life. Cells were a mystery. Chromosomes were a mystery. Genes were a mystery. The world needed a breakthrough. In the 1940s, Oswald Avery and his team discovered that DNA, not protein, carried genetic information. It was a shocking idea. Most scientists didn't believe it. How could something so simple contain all the instructions for life? The answer would come from 2 young scientists who saw what no one else could. In 1953, James Watson and Francis Crick proposed the structure of DNA, the double helix. 2 strands, twisted like a spiral staircase, held together by base pairs that fit like puzzle pieces. A structure that could copy itself with astonishing accuracy. It was elegant. It was simple. It was revolutionary. Suddenly, scientists understood how information was stored, copied, and passed on. Biology became predictable. Life became readable. The age of molecular biology had begun. Once the structure of DNA was known, breakthroughs came rapidly. Scientists uncovered how DNA codes for proteins, how genes turn on and off, how mutations cause disease, how viruses hijack cells, how cells repair their own DNA. For the first time, medicine had a molecular explanation for illness. Sickle cell disease, cystic fibrosis, hemophilia, cancer, genetic disorders. They all had molecular signatures. And now, scientists could find them. In the 1970s, a new idea emerged, not just reading DNA, but editing it. Scientists discovered restriction enzymes, molecular scissors that could cut DNA at precise locations. This led to recombinant DNA technology, the ability to combine DNA from different organisms. It was the birth of genetic engineering. Suddenly, bacteria could be programmed to produce human proteins. Viruses could be modified to deliver genes. Cells could be reprogrammed to behave differently. Biology became programmable. This moment created an entirely new industry. The 1970s and 1980s saw the birth of companies built entirely on DNA technology. Genentech, Amgen, Biogen, Genzyme. These companies weren't chemical manufacturers. They were molecular engineers. And one company would prove just how powerful DNA technology could be. In 1976, a small startup in California called Genentech set out to do something no one had ever done before: use genetic engineering to create a human medicine inside bacteria. Their goal was bold. Their resources were tiny. Their idea sounded impossible. At the time, people with diabetes relied on insulin extracted from pigs and cows. It worked, but it wasn't identical to human insulin. Some patients had allergic reactions. Supply was limited. Demand was rising fast. Genentech's founders, Herbert Boyer and Robert Swanson, believed they could engineer bacteria to produce pure human insulin, not animal insulin, not synthetic insulin, human insulin. To do this, they used recombinant DNA technology, cutting human DNA, inserting it into bacteria, and turning microbes into microscopic factories. In 1978, they succeeded. For the first time in history, a human protein was produced in a living organism that wasn't human. It was a scientific earthquake. Genentech partnered with Eli Lilly to scale production, and in 1982, the FDA approved Humulin, the world's first genetically engineered drug. Humulin became a blockbuster. It transformed diabetes care. It proved that biotech wasn't science fiction. It was the future of medicine. And it launched an entire industry. Suddenly, biotech companies weren't just startups. They were the new giants of medicine, capable of creating therapies that chemistry alone could never produce. In the 1990s, scientists launched one of the most ambitious projects in history, mapping every gene in the human body. The Human Genome Project. It took 13 years. It involved thousands of scientists. It cost billions of dollars. But in 2003, the human genome was completed. For the first time, humanity had a full map of its own genetic code. This achievement opened the door to personalized medicine, genetic testing, targeted therapies, precision oncology, pharmacogenomics, gene therapy. Medicine was no longer one size fits all. It became personal. The DNA revolution reshaped the entire pharmaceutical landscape. Companies shifted from chemistry to biology. Drug discovery became data-driven. Diseases were understood at the molecular level. New drug classes emerged: monoclonal antibodies, recombinant proteins, gene therapies, RNA-based medicines, CAR T-cell therapies. This era laid the foundation for everything from mRNA vaccines to CRISPR gene editing. Biology became technology, and technology became medicine. The DNA revolution teaches us that the biggest breakthroughs often come from understanding the smallest details: a molecule, a code, a sequence. Once we understood DNA, we gained the power to diagnose, treat, and even prevent disease at its source. This era didn't just change medicine. It changed what medicine could become. If you want to keep learning about the incredible world of medicine and build the skills to thrive in one of the fastest growing industries on the planet, make sure you like this video, leave a comment, and subscribe to ScientificCareer's. Your journey in science and healthcare is just beginning, and we're here to guide you every step of the way. But unlocking the code of life was only the beginning, because once scientists learned how to read DNA, they wanted to learn how to rewrite it. In the next episode, we explore the rise of genetic engineering, monoclonal antibodies, and the birth of modern biotech therapies, medicines designed with precision, built from living cells, and targeted at the molecular level. Episode 7: Engineering Life. The rise of modern biotech and targeted therapies. Don't miss it.